I'm Katrina Rokosz. I'm part of Yang Chen's lab here at Stony Brook University. And this project is kind of a branch off of what Xiaoyun just presented on, focusing on that diel period variable. So for those of you that just joined us, I'm going to give an overview of the Hudson River Biological Monitoring Program. For those of you that have been here, it's a lot of repeat of what you've just heard. But the HRBMP is a survey program that consists of 6 surveys, including the Long River Ichthyoplankton Survey and the Water Quality Survey. The data that I will be using spans 43 years, from 1974 to 2017. And the survey collects data for all of the Hudson River estuary, from the Battery in New York City all the way up to the Troy Dam north of Albany. The Hudson River estuary is a pretty dynamic system, with the lower estuary being more of a marine system, the upper part of the estuary being more of a freshwater system. The water quality survey data consists of temperature, dissolved oxygen, and conductivity. And that data is collected at fixed stations throughout the Hudson River estuary at 3 depths. The data that I will be using will be from the LRS, the Long River Ichthyoplankton Survey. This is a survey, it's a ichthyoplankton trawl survey that collects data on length, abundance, and life stages for over 170 fish species in the Hudson River estuary. It has a stratified random sampling design with the Hudson River estuary broken up into 13 regions, which you can see in this figure here. And 2 gears were used, as Xiaoyun mentioned, the Tucker trawl and the epibenthic sled. And over the course of the LRS, there were over 108,000 tows that were done, combining the 2 gear types. There are other there's a lot of protocol changes. One of those is the diel period, which Xiaoyun touched on previously. This was not a random variable in the survey design. It was consciously chosen every year when they would do day versus night sampling. So why ichthyoplankton matter and why this survey is so important. Ichthyoplankton egg abundance is related to spawning stock biomass, and ichthyoplankton are indicators for changes in fish community. Early life stages are critical for recruitment, and larvae mortality is influenced by environmental variables. And these pictures here are taken by Patty in our lab, and they're from the biological collection that's part of the HRBMP. So why this diel period, part of why it's such an issue is that older ichthyoplankton have been seen to engage in gear avoidance, and larva fishes have been found to engage in diel migrations. So these concerns spark the question of how does relative abundance represented by sampling that was done during the day compared to relative abundance of sampling conducted at night. And so striped bass and white perch are pretty important. Striped bass, I've been told, is king in New York. So I looked at the relative abundance of striped bass and white perch over the study period, separating it between day and night. And there's a lot of variables in the Long River survey, and so I isolated the data to just be, this is summer data collected during the summer for yolk sac larvae for striped bass and white perch. And this is between the 2 gear types that were used. And you can see that the pink here represents the daytime relative abundances, and the blue represents nighttime. You'll notice a gap in the pink day relative abundance, and that's because daytime sampling was not conducted during these years. And you can see some pretty interesting trends. The white perch relative abundance is a lot higher than during the day compared to at night. And the opposite seems kind of true for striped bass. So I wanted to see if in the whole community of the Hudson River estuary, using the LRS data, there are trends in which certain species dominate the catch during the day compared to at night. So what I did was, again, I subsetted the data, just in this case, just looking at yolk sac larvae collected in the summer months, which were May, June, and July in this case. And since the Hudson River estuary is so dynamic, this is just looking at the freshwater portion of the Hudson River estuary. And I calculated relative abundance for all the species in our data has, and took the daytime relative abundance and subtracted the nighttime relative abundance. So if there isn't a difference in daytime versus nighttime relative abundance, community is the same, then this figure would be empty, it'd be blank. But there are differences which you can see. And you can see that the white perch and this dark maroon, they're dominating way more during the day, whereas striped bass at night have a higher relative abundance compared to their daytime relative abundance. So the figure I just showed you, that's now on the top panel up there, and so then the top panels show the relative abundance calculated the same way I just described for the upper Hudson River, the lower Hudson River, and then since we did use two types of gears, I separated it between sled and trawl. And so this pattern is clear across gear type and region where there are differences in what species are caught during the day in our data versus at night, with white perch dominating the daytime relative abundance and striped bass dominating at night. So for the rest of this analysis, I've focused on striped bass and white perch because they are the dominant ones. And just a little background, striped bass and white perch both spawn between April and June in the Hudson River. They're both freshwater spawners, preferring freshwater regions of the Hudson River. Striped bass eggs are pelagic and buoyant, whereas white perch eggs are demersal. And so the overall objective that I am aiming to address is just to assess the differences in relative abundance indices for striped bass and white perch during day, data collected during the day versus at night. And so there are 4 questions that I was aiming to answer. The first is, are there differences in relative abundance between the different diel periods? Then do different variables impact the relative abundance represented in the data collected during the day compared to at night? Is diel period a significant variable in predicting relative abundance during these different diel periods? And what would be represented in the data if we just completely took out diel period, combined all the data, and treated it like it wasn't a factor? So what I did first is just a simple t-test to see if there are significant differences in daytime and nighttime relative abundance. Because there's so much data, this was done on the species level and the life stage level. So striped bass and white perch eggs, yolk sac larvae, and post-yolk sac larvae. After that, I ran generalized additive models, or GAMs, to figure out what variables are significant in predicting relative abundance using data from the daytime and nighttime and combined. So looking at the t-test, these are the p-values, striped bass, eggs, yolk sac larvae, post-yolk sac larvae, all significant. Interestingly, post-yolk sac larvae for white perch, when you just aggregate everything, was not significant. These are the figures calculated the same way as what I previously showed, but this is just for post-yolk sac larvae, and what you can see is this temporal change in when white perch post-yolk sac larvae are dominating in the upper Hudson River in the earlier years. It seems like at nighttime their relative abundance is higher, and then it switches to daytime. And that break, I think I forgot to mention, that break in the data is just because, again, daytime sampling just wasn't done. So the t-test just reflects, the results of the t-test just show that this data is so complex and these finer scale patterns, you can't just aggregate it. So when you break up the data into more of a finer temporal scale than just all of it together, there is significant differences in day and nighttime relative abundance for white perch. Onto the GAMs. What I did for the GAMs is they were run to determine what variables are significant in predicting relative abundance. And so for each species and each life stage, I ran 4 models. The first one was just using data from the daytime tows for relative abundance. The second was just using data from nighttime tows for predicting relative abundance. And then the last 2 models were if you combine the daytime and nighttime data all together. The first combined data model is if you didn't include diel period as a predictor variable, what variables are shown to be significant. And then the 4th model is if you do include diel period as a predictor in relative abundance, does the model change? So this table is just to show you the output. I know that a lot of people may not be familiar with GAMs. But the point of this is to show, the first column is post-yolk sac larvae, that just shows you the life stage The 4 models are represented in the second column, and then the remaining columns are all predictor variables that were included in the model. This is not the complete list of predictor variables that were included in my model. Variables including year and river region, they were also included, but the variables that were significant in every model run were left out of this table just for simplicity because there's a lot going on here. Then the colors represent different ways that variables were inputted into the model. So the red no just means that this in no way, after the model has been tuned and the best fitting model has been found, that variable was not included in the model. It's not a significant factor in predicting relative abundance. The yellow-orange interaction term means that that variable, when put in the model, is not included by itself. It's only included when it's interacting with another variable. So in this case, conductivity is only included when interacting with temperature, and same with dissolved oxygen. The smoothed means that that variable is included in the model, but a smoothing term has been applied to it because it has a nonlinear relationship with relative abundance. And then yes just means it's a significant factor and was included in the model. So you can see here that the 4 models, when you use different data that has different diel periods included in it, the predictor variables are different. And for that 4th model, in every single case in which it was included, in which diel period was included, it was significant. So that's also why it's not in the table. So this is all of the models I ran for striped bass. And so what you can see is for yolk sac larvae and post-yolk sac larvae, for depending on what data and what diel period that data came from, the inputs of the models are different. But interestingly, for striped bass eggs, the model inputs what's significant in predicting the egg abundance is pretty much the same with the exception of conductivity for daytime data. This is the model outputs for the white perch. Across the board, all of the inputs are different. And so the overall conclusions I want you to get from these GAMs is that when included as a predictor variable, diel period is significant in predicting relative abundance across both species and all life stages. And the predictor variables that are significant for the best fit models, they vary depending on what species you're looking at in life stage. So therefore, diel period should not be overlooked when looking at relative abundance, or when doing relative abundance analysis. So back to those questions that I had at the beginning. That first question of, is there a difference in relative abundance using data from daytime versus nighttime catches? Yes. Do different variables impact abundance during the day compared to at night? This depends on what you're looking at. If you're, in the case that I just showed, striped bass eggs, the variables don't really change between daytime versus nighttime data, but for all the other life stages and for white perch, the variables do. Is diel period a significant predictor in relative abundance? Yes, when the data was all combined in every single model run, the diel period was a significant predictor variable. That last question, which is what would be represented in the data if we ignored diel period and just combined everything, I haven't gotten to yet. That's the next step in this research. I want to see what data should be used, under what circumstances, if any, can we combine daytime and nighttime just to simplify this really complex data set a little bit. So the overall take-home message is generally diel period should not be overlooked regarding relative abundance for species and early life stages. We need to be careful with the Long River survey data, a little bit messy, and there's further need for in-depth analysis to allow us to really understand these inconsistencies, not just these, but the ones Shaggy also presented on in the Hudson River data, and develop an approach to overcome these problems. I just want to acknowledge Entergy for the gift of the Hudson River Biological Program data, the Hudson River Foundation, the New York DEC, SOMAS, and the Hudson River HRBMP group that I've been working with. Any questions?